you get effects in the genomes where genes cooperate with each other, and you get gene complexes. One of these gene complexes is called the major histocompatibility complex, which controls your immune system. And the other gene complexes where you don't inherit just a single gene, but you inherit a set of genes that work together. And then these are likely not to follow the second law. Okay, so we're going to demonstrate an experiment that Mendel did that's on incomplete dominance and illustrates his first law, which is random segregation. And he did this with snapdragons. So what Mendel did is he selected two lines of snapdragons, and one consistently only produced white flowers, and one consistently only produced red flowers. Then what he did is he took one from each of these lines and he crossed them, and he got what's called the F1 generation. That's the first progeny generation. And the individuals in this generation were all pink. So imagine you're Mendel and you do this experiment. You don't know anything about genes. You just know that here's a set of flowers that for all time going backwards, they've always produced red flowers. And here's some plants, they've always produced white flowers. And you notice that sometimes they interbreed and you get these hybrids that are pink. So you do this experiment and you take a plant that only produces red and a plant that only produces white, and you cross-pollinate them. And you can do this physically by just with a paintbrush, taking pollen from one and brushing it onto the stamen of another. And then you notice that all the plants that arise from the seeds of that cross are pink. And now you take one of these pink plants, let's say this one, and you cross it back with the white. And now what happens when you do this is half of them are pink and half of them are white. But if you take two pink, then a quarter of them are white, half of them are pink, and a quarter of them are red. So you do these experiments and you sit there and you say, wow, this is weird. I'm getting these fractions, and it's almost, certainly there's going to be some experimental variation, but if you do enough flowers and you do this enough times, these fractions get closer and closer to a quarter, half, a quarter in this case, a half, half in that case. So you sit there and you say, how do I solve this puzzle? So the simplest way to solve this puzzle is to postulate that each flower has two alleles at the gene for flower color. And white flowers have these two alleles that we'll call little r and little r. So it's homozygous for little r. You can call them WW, you can call them whatever you want. So historically, if you look in textbooks, people have called them different things. And the red one we'll call capital R, capital R. So if you're red, you're homozygous and you have these two alleles for redness. And if you're white, you're homozygous and you have these two alleles for whiteness. And then when you cross, each white flower donates a little r, and each red flower a capital R to the progeny. So all the progeny have the alleles capital R, little r. And now, if these have blended inheritance or are co-dominant, you get a blended color, and the blended color is pink. So that sort of works nicely. And let's see if we can explain the rest. Well, if you take two pink flowers and you say, okay, I look at a pink flower, and I'll call this big R, little r, big R, little r. And so this will make a bunch of zygotes, and half of them will be big R. So these are making all these haploid pollen or ova that are going to be fertilized by pollen, and half of them are big R, and half of them are little r. If I choose any one of these, I can either get a big R, or if I choose 4, I'm likely to get a big R and a big R and a little R and a little r. If I choose 4 here, I'm likely to get a big R and a big R and a little R and a little r. Now, obviously, there's going to be some variation because it's like throwing a dice. So we're just talking about averages for large sample sizes. Now, when these come together, this big R could either get a big R or it could get a little r, and this little r could either get a big R or a little r. So if you have a look at it, you expect a quarter of the time you'll end up with big R, a quarter of the time you'll end up with little r, and sometimes you'll get big R from one parent, little r from another, or little r from the first parent, big R from another. So you can say, if that's the father, that's the mother, then you can get a big R or a little r from the father, and a little r or a big R from the mother. So half the time, you'll get that combination. Now, it makes no difference whether it's big R, or little r, big R. That's going to be pink. So half the time it's pink, quarter red, quarter white. Everybody understand that? And if you confirm that, then you would predict that if I took a pink with a white, from the pink I can either get a big R or a little r, but from the white I can only get a little r. So half the time I'll get big R, little r, and half the time I'll get little r, little r. So half the time I'll get pink, and half the time I'll get white. So when Mendel did these experiments, this is what he worked out, and he was very happy. And he said this is the simplest possible explanation for why these particular phenotypes emerge with this kind of crossing. Okay? So then he did a more complicated experiment. And what he did is he looked at pods from the sweet pea. And he found out that pods could either be green or yellow, and they could either be smooth or lumpy. So he has a smooth green, smooth yellow, green lumpy, yellow lumpy. And when he did this kind of experiment, he found out that he would start out with peas of a particular kind, and he would keep breeding them until, let's say, he had only this kind, and that is yellow and smooth, or green and smooth, or green and lumpy. So now, if you start out with two individuals that are heterozygous for both these traits, big R, little r, big Y, little y, and again, the mother is big R, little r, big Y, little y, and the father is big R, little r, big Y, little y. If you go through all the combinations that you get, you can either get a little r and a little y from the father, and a little r and a little y from the mother, and so this would be here from the mother, and this would be what you can get from the father. So from the mother you can get little r, little y, big R, big Y, big R, little y, big R, little y, little r, big Y. And you can get the same combinations in the zygotes from, or the gametes from the mother. And then when these come together, they'll give all these different combinations. Now if big R dominates and big Y dominates, then only one of 16 cases can be yellow smooth skin. Because whenever you, in order to get a yellow, you need two small Ys. And in order to get a smooth skin, you need two small Rs. The rest of the time, you're going to get the big R dominates the little r. So whenever you've got big R and little r, you're going to get lumpy skin. And the big Y dominates the little y, so that whenever you big Y, you'll be green. And whenever you've got a big Y. So here's big Y, little, sorry, it's the other way around. The big Y dominates, the big Y gives you, this doesn't look right. Okay, yeah, sorry, I got it. Yeah, the Ys are for, the Rs are for color, and the Ys are for, yeah, these are not great symbols. But anyway, if you go through this combination, then you'll see that you need segregation and independent assortments. In other words, the R that you get and the Y you get are not linked. And you can equally as likely get a big R or a little r if each parent has both. And then when you work out all the combinations, then this is the table that you'll get. So these sorts of ideas lead to simple genetic models that we build. And these models are called, they're simple models, and they're called multi-allele, multi-locus models, and the simplest of them is a single allele, a two-allele, one-locus model. And that's the model that we'll look at now. And so let's set up a very simple genetics model. So we now consider a model of how a particular quantity changes from one generation to the next. And so we can say, let X, if we have a model where we can have two alleles, big A and little a, and we look at the population. So in the population, we have some individuals that are big A, some individuals that are big A, little a, big A, big A, little a, little a, little a. And when I say big A, little a, and little a, big A like this, if I don't care which one comes from which parent, then these are the same. It's only if one comes from the father and one comes from the mother that this turns out to be important. But in most cases, it doesn't matter which gene you got from which parent. It doesn't make any difference. So for eye color, if you've got a particular eye color, it doesn't matter whether you've got the combination in one way or in the opposite way in terms of your parents, you still have the same eye color. Now this is not entirely true for all genes, and we'll come back to this. But if I take a population of individuals, and I take all their particular genotypes, and I have a look at the gene pool, then I can say the frequency of a particular allele is X. And then the frequency of the other allele is going to be what? If I've got only two possibilities, big A and little a, and the proportion of big A's in the population is X, what's the proportion of little a's? 1 minus X, okay? So this is the simple basis on which we build these models. So we have these genotypes, big A, big A, little a, big A, little a, big A, big A, little a, and big A, big A. And the model that we build is only valid for large populations. And the reason why it's only valid for large populations is that these random segregation rules only can be accounted for by proportions when a population is large. So for example, if I have a coin, and I flip the coin, and I say to you, I do this many, many, many times, and I say to you, what proportion of times will I get heads, and what proportion of times will I get tails if it's a fair coin? What will the proportion be? 50%, so it'll be a half. Now, if I do it twice, I could get 2 heads or 2 tails. So I can't say if I do it twice that the proportion is going to be a half. It's only if the population size is large enough, so I've done this thousands of times, that I can say the proportion is a half, and within sampling error, it may be 0.499 or 0.496, depending on how big the sample is. But it approaches a half as the population gets big. So all these models, because I'm not going to work with probabilities, I'm going to work with proportions, assume that I'm working with large population size. So there's the random mating principle, and the random mating principle says that if the frequency of little a is X, and the frequency of big A is 1 minus X, then the frequency of this particular individual, this genotype, is going to be equal to what? If I'm drawing one of these from the population, I draw it with probability X. And if I draw it again, it's probability X, so it's X squared. If it's little a, big A, or big A, little a, I can draw this in 2 ways. I can either draw little a with probability X and big A with probability 1 minus X, or I can draw big A with probability 1 minus X and little a with probability X, and this will give me 2X times 1 minus X. And then, if I have a look at what's the probability of getting this particular genotype, AA, what's the probability of drawing A, which is 1 minus X, and A again, which is 1 minus X squared. Now, if you add these up, X squared plus 2X times 1 minus X plus 1 minus X squared, what is this going to be equal to? 1. So you can show that everything cancels out and you're left with 1. So that these are going to be the different frequencies. So now let's talk about a model. So let's say the allele A is what I call a lethal recessive. So what do I mean by a lethal recessive? What I mean by that is that if you have 2 of those alleles, you don't make it. If you have a little, so that means if you, this genotype, you don't make it. If you're this genotype, you're fine, and if you're this genotype, you're fine. So let's see what happens if we have a certain proportion of A in the population. So I'm going to say X at time t, or I can use N. Let's see if I use N. Xn represents the proportion of A at nth generation. So now I want to see what happens in the nth plus 1 generation. So I say, okay, what's xn plus 1 going to be? Well, if we reproduce the population many times, and if all these individuals die, but all these individuals live, then basically, from these individuals in the next generation, there'll be no contribution to the proportion little a. And from these individuals in the next generation, there'll be a contribution of half of them will be little a, half of those individuals, and half will be big A. So if you do this, then now if it's easier for you, you can say, suppose that I have n of these, 2n of these, and n of these, and the n's will cancel out. So it's equivalent to saying that if I take these individuals, half of their alleles contribute to little a in the next generation, and then what's left, these individuals are not going to be there, so I'm going to have 2x, 1 minus x, plus 1 minus x squared. So I'm going to have all these individuals plus all these individuals, none of those individuals. So the total number of individuals in the population will be this plus this. But the proportion of those that contribute the allele, little a, will only be half of this contribution from this group of individuals. And so now if I simplify this expression, I'll get, you can work through it. So here's the expression. I've got the n's in, the total number of alleles. The n's cancel out, dividing top and bottom by 1 minus x over n, and that simplifies to xn plus 1 is equal to xn over 1 plus xn. So I have a simple expression now that tells me how the proportion of alleles, little a in the population, will move from one generation, will change from one generation to the next. So I can make a model, I can make a ANOVA model, and if I start out with half the alleles being these lethal alleles, then it'll drop very rapidly, and after 100 generations, there'll be almost no alleles left of these lethal alleles. Now it turns out that biologists did an experiment with Drosophila, and they found that in Drosophila, when they bred them, they would get a mutation. And this mutation would be called glued, G-L-U-E-D. And glued, it's called glued because it would make the wings stick together, and these individuals would never reproduce, they would die. And so if you start out in a population where the proportion of glued genes in the population is a half, then you can see that each generation, the number of glued genes drops in this kind of exponential fashion. It's not quite exponential because it's not as fast. And so this is the theory, and then this is what they found in practice. So you can make a simple model of what you'd expect to happen to these lethal genes in a population of Drosophila, and you can do experiments, and you can have the theory, you can have the model, and then you can test the model against the theory. So now, this is a very simple situation where the gene is lethal. It's a lethal recessive. But typically what happens is that genes are not necessarily dominant or recessive, but they may act together, or the alleles may act together, and they may not be absolute. In other words, if you have a look at the situation, and you say, okay, little a, what we can do is we can assign relative fitnesses to these different phenotypes or genotypes. So we can say if we have big A, little a, that's the heterozygous. And we can say this has a fitness of 1, a relative fitness of 1. Then we can say little a has a relative fitness W1, and big A has a relative fitness W2. So these are just parameters, W1 and W2. If W1 is greater than 1, then we say these homozygotes are fitter than these heterozygotes. If W2 is less than 1, then we say these homozygotes are less fit than these heterozygotes. So we can get different relationships. We can get W1 is greater than 1 is greater than W2. And that just says that the allele A, if you have the allele A, you're going to be fitter than if you have the allele big A. Little a gives you greater fitness than big A. And not only that, if you have 2 copies of it, you're fitter than if you have 1 copy of it. You can make W1 equal to 1 greater than W2. Then you can say that big A has the effect of reducing your fitness, but only in the recessive state. So there's all these different combinations. Now the 2 interesting combinations that arise, one is W1 is greater than 1, and W2 is greater than 1. So this is, let's say, case 1, case 2, case 3. And here, W1 is less than 1, and W2 is less than 1. So in the second case, what it means is the homozygote is fitter than the heterozygote. It doesn't matter what kind of homozygote you are, you're going to be fitter than the heterozygote. And this is the case that's called disruptive selection. And in disruptive selection, what you'll show in your homework